we start talking about some of these, specific infections, I want y'all to have a little bit of a grasp about our response to those infections, so those response to infections. How many of y'all had any knowledge of your Get cozy. You're going to be bored. So immunology, study of immune responses. Immunity is the ability of us, so we are the host, to resist a particular disease or infection, and the immune system is all the components of our immune system that actually aid in fighting off these different kinds of infections. We'll talk about some different things that can happen, but your immune system can recognize all kinds of things as foreign. And sometimes when you have an autoimmune disease, you can even recognize things that are part of you that are not foreign, but it will mistake them as foreign. And you'll have an autoimmune disease then. And there are different lines of defense. First, second, third. First is really basic. So the first line of defense is going to be non-specific. So it's going to be something that pretty much everyone has that just blocks out the infection somehow. So something like skin. You're going to have epithelial cells on your skin. That's going to prohibit organisms from getting into your body. So non-specific. Second line of defense is going to be protective cells and fluids, inflammation, phagocytosis, so also non-specific. So different kinds of protective cells. We'll talk about this a little bit, and I just talked about it here a couple seconds ago. Your epithelial cells on your skin, as they, I'm not talking about this in here, as they age, they fill more and more with keratin. And that keratin, fully keratinized epithelial cells, when they get filled all with keratin, they fall on your body. With them goes organisms or vascular. So there's a constant turnover. That constant turnover aids in precluding pathogens from attaching to you. Fluids. There's different ways you can look at fluids. It's the second line of defense. Your body has a lot of kind of innate kind of responses to infection. A lot of that involve fluids. So constant flushing. So crying, urinating, constant flushing action. I'll give you an example of urinating here. So sometimes, I think I talked about that here too. Sometimes you'll have an infectious disease doctor that comes across the urine specimen and has E. coli in it. And a lot of people will be like, we got to get antibiotics to have E. coli infection, they have E. coli UTI. And if those patients aren't symptomatic, so they don't have the symptoms of UTI, a lot of doctors don't treat it anymore. Because there's a certain kind of E. coli that makes a villi that allows it to attach to your urogenital tract that allows it to establish infection. If the E. coli happens to be an E. coli that doesn't have those villi, when you eat a lot, it's going to eventually flush them out. So that's a flushing action. Crying, vomiting, all those things are kind of these innate responses to get bad things out. Sneezing, coughing, having diarrhea. We won't talk about all those in here, but those are all things to get things out of. Inflammation, we'll talk about inflammatory response here a little bit. Throughout this lecture every day. Phagocytosis, a lot of phagocytes, so white blood cells that engulf pathogens for destruction, presentation to other cells in the immune system. Those phagocytes are non-specific, so they just recognize pathogens because they recognize LPS or they recognize the component of Flagella or something like that. They have, or maybe you've heard of the term toll-like receptors, right? So phagocytic cells like macrophages have on their surface these toll-like receptors. These toll-like receptors recognize components of pathogens. Bacterial DNA, Flagella, LPS, things like that. But it's non-specific. It's not, those phagocytes aren't specifically recognizing a specific strain, Streptococcus. They're recognizing general stuff, generalized forms. So non-specific. When you get into the third line of defense, then you're talking about things like B cells, T cells, things like that. But they are recognizing something very specific. That is because they're recognizing something you've likely been exposed to before and have a little bit of an immunity against. So we'll talk about how that happens too, how your immune response happens. Above the black line, good things. Below the black line, bad things. So some good things about your immune response. Gives you defense against infections. Does that always work? No, unfortunately it does not. Or I wouldn't have a job if we wouldn't be here today. But a lot of times it prevents you from coming out with these different kinds of diseases for sure. Defense against tumors. Your immune system has the ability to recognize cells that are atypical, meaning they're malignant cells, cancerous cells. Obviously that unfortunately doesn't work all the time either. Implications if something goes wrong with these different defense mechanisms for infections and cancer, deficient immune response, and you have susceptible to these different types of cytokines. You can exploit some of these immune responses to actually treat cancer. So that's what we talk about immunotherapy. I don't think, when's the last time, nobody here's had cancer biology, right? I think it's been a long time since Lindsay's on that. So Lindsay Rhodes, the farm chair right now, she has a cancer biology course. I think it's been a minute since she's on her. But that's some of the stuff that Lindsay talks about. The bad things. Immune system can injure cells and induce pathologic inflammation. So essentially that means your immune response is out of control. One of the examples I'll likely give you are either as I continue to read what they do is sometimes you have pandemic strains of the flu. This is the example I gave you. Those pandemic strains are usually strains that have interacted with a flu virus and an animal. It might be swine, a pig, avian, bird. And what happens is you get kind of this hybrid influenza strain that has some characteristics of an animal influenza virus and some characteristics of a human influenza virus. Your immune system does not know what to do with the half bird virus, and it gets really confused and it overreacts. So essentially, when you get this hyperactive inflammatory response, it ends up damaging you as well as trying to clear virus. That's why with pandemic strains, I'll ask you this. I'll think we're talking about this. You have, let's say you have three groups of different ages of people. You have very young, middle-aged, and elderly. So you have, if you're talking about a random type of infection, which of those populations do you think usually gets the sickest? The really young, and or the middle-aged people, and or the older people?
The really old and
The young and old, right? Why is that?
because they're more immunocompromised.
Because they're more immunocompromised, right? Those pandemic flu strains is usually the middle-aged people because they have the strongest immune response. So their immune system overreacts, and they have, I don't want to say more immune system, but they have a more robust immunological response. So when you start to see the middle-aged people getting sick from something, that's bad. We started to see that with some of the strains of COVID-19. Remember when we were going through the pandemic and the heat of that? I think maybe a lot of you are probably in high school for that. God damn, I'm old. Does anybody remember what they used to treat COVID-19 patients with to suppress their immune response? Paxlovid, but what did they give them to treat the immunological? They gave them antibiotics, so sometimes passive immunization. Sometimes they took plasma, yes. But what did they use to treat the immunological response? They gave them one of these therapies to treat the infection, but they would also give them steroids. A lot of people, especially the ones that were having like really severe like breathing difficulties, sometimes they give them steroids as well. They're getting steroids because a lot of the inflammatory response is an overreaction for COVID thing, for the SARS-CoV-2 virus, so they give them steroids too. So you get a package, you get something. You get something probably in maybe in some antibodies. You got something, and then you got steroids. That makes sense.
You say rates help reduce like overreaction.
Yeah, so it's someone something that's suppressing their immune response while they're actively trying to use their immune response to fight off infection they have. So it's a dangerous game. You usually don't play that game unless you don't have any other choices left. And for those of you, for all of you, because you're alive and none of you are like 3, for all of you, when you went through that pandemic, that was the crap pit thing. So that's what we do. As the immune system recognizes and responds to tissue grafts and communicates with the protein. And so we kind of already started talking about this a little bit in a way. You give someone a kidney transplant. Well, that kidney's foreign to them. So their immune system's going to recognize that organ as foreign. And the immune system's going to start attacking it, which is going to result in organ rejection. So you give them you put them on immunosuppressive therapy so their immune system doesn't bring that over. If you do not do that, it's transplantable. So the good and the bad. Alright. So your immune system, 2 arms, the innate, which is the first and second line that we talked about a little bit, and then your adaptive, which is the 3rd line. So your innate immune response is native or natural immunity, which means everyone's got it. You don't have to be exposed to something that has this type of immunity. Everyone should have macrophages. Everyone should have BMMs, which are neutrophils. Everyone has skin. Hopefully. Those are things that are innate. Adaptive exposure to something, some kind of a pathogen, gives you an immunity. So your immune system has an immunological memory. So that's your 3rd line of defense. So acquired, sometimes you see this referred to as specific immunity. It's the same thing. Adaptive, acquired, specific, it's all talking about the same thing also. That adaptive immune response is going to include your antibody-mediated humoral immune response. So things are called antibodies, referred to as humoral immunity, and your cell-mediated immune response, which you use to talk about T cells there. B cells make antibodies, plasma B cells specifically, and T cells are the cell-mediated response. So we'll talk a little bit about how they happen. For those who had immunology, we are like very, like 120,000 foot view. So, like I said, get cozy. Won't be bored. Okay. So here are kind of the players you have for the innate and adaptive immune response. Innate immune response, you're going to have phagocytes, so your macrophages, dendritic cells, neutrophils, things like that. Components of complement, which we'll talk a little bit about. For those of you that had immunology and had to remember all the parts of the complement cascade, I don't think you know that in here as well. Don't tell, sorry. And your natural killer cells, we won't really go into those too much. Adaptive immunity, you have B cells, which are involved in antibody production, and your T lymphocytes. So things like upper T cells, sometimes T cells, things like that. So let's talk about the first line of defense first. First line of defense against microbial invaders is going to be your innate immune response, because again, you have that present intrinsically, so everyone has it, it's innately there. It's not specific, so it's going to be a rapid response because everyone has it. This is when we start talking about things like opportunistic infections a little bit more. Again, part of your innate immune response is something like your skin. Well, if you have a break in your skin, something because of that wound causes an infection. So that would be a break in your innate immune response through one of these intrinsically present barriers. Lacks immunological memory. Your skin's not going to remember if it came across some kind of a pathogen last week. It's skin, and there's no way for it to do that. Your respiratory tract has mucus and things like that. The components of mucus, I'm not going to remember what it was exposed to. There's no memory. Limited diversity of expression and limited number of pattern recognition receptors. So pattern recognition receptor, they're talking about a toll-like receptor. It's going to recognize general things for pathogens, but not specific pathogens. Algae, flagella, components of peptidoglycan, things like that most of these bacteria are going to have, but they don't have, so it's not going to recognize it. Those toll-like receptors is going to be able to tell us between chlamydia and staph. It's just looking toll-like receptors, that's what those are. They're gonna recognize pathogen-associated molecular patterns, or PAMPs. So that's what I was just talking about, those kind of general things that all these pathogens are gonna have. Sometimes you'll see PAMPs referred to as that you take your MCAT, you take DAP, whatever, or you take something on down the road, some kind of board exam. Sometimes, depending on the age of the individual writing the exam, you'll sometimes see these referred to as PAMPs. It's the same thing. So instead of pathogen-associated molecular patterns, it's a microbe-associated molecular pattern. It's the same. But sometimes you still see PAMPs used instead of PAMPs. If I remember correctly, I believe that's how I was trained, PAMPs. It's the same stuff. They can also sometimes recognize damage-associated molecular patterns, or DAMs, and those are artifacts from damaged cells in your body from infection itself. We'll focus more on PAMs and DAMs, not so much on DAMs. We'll touch on a little bit. Anatomical barriers, physiological, chemical mediators, phagocytes, complement, inflammatory events are all things that are of your daily immune response. So we'll walk very slowly through all those with different examples there. Starting with the skin and mucous membranes, the respiratory tract, your general eyes and digestive tracts. Again, outermost layer of your skin has got the epithelial cells, can have a bunch of keratin into it. A lot of pathogens can't penetrate through that. There's a constant turnover of those cells as well. And you have normal flora on those cells. Flushing affects the sweat glands. As fluids are flushed, they're going to flush things out. Damaged cells are going to be replaced. Mucus coated yeast attachment and the entry of bacteria. So if you have a pathogen that you say they don't break, but is trying to establish an infection in, say, the respiratory tract. Well, there's a lot of bacteria that can't actually get through that thick mucin layer in your respiratory tract, actually attach to the underlying respiratory epithelium. Like it can't get through that. There are other bacteria that make different enzymes that break down the mucin, that potentially can use the mucin as a carbon source. Those are the ones that are better at establishing infection in the respiratory tract, because they can penetrate through that mucus. The other thing is a lot of those fluids, like mucus in your respiratory tract, is going to have different enzymes, things like lysozyme. So lysozyme is an enzyme that breaks down peptidoglycan, and bacteria have peptidoglycan. So it's hard for a lot of bacteria to get through there because there's things that kill them. Linking and tear production, so say you have some kind of an eye infection, that can help flush that pathogen from your eye. Stomach acid, we talked a little bit about, it's got a low pH around 2. Most microorganisms can't survive that, so if you eat something, it can potentially cause a gastrointestinal tract infection, and it goes through your stomach. Most times, your stomach, if it's a small amount, the bacteria is going to be killed with a low pH. The ways that most of those, we don't talk about GI infections in here, we do that in the spring, but the way most of these pathogens actually survive through your stomach is there's just a ton of them, so you just consume a lot, or it's in some kind of a tube, it's a big one too, so you have microorganisms, and it's in a big like clump of food, and the food doesn't get broken all the way down your stomach before it passes through your small intestine. So that food actually protects the bacteria from that low pH. This is why we talk in the spring, for those of you that have the spring. If you look at the infectious doses for GI pathogens, or if they're consumed in water versus consumed in food, the dose usually has to be a lot higher in water than what it is in food. Because if you just consume something like cholera in water, there's nothing protecting that, it's just water. So they get into your stomach, there's nothing shielding it. So the item then changes infectious doses. Again, we'll talk about that next semester. Nasal hair traps, larger particles, y'all understand that. And again, microbial antagonism, like normal flora. I don't think I've used that specific term here yet. Microbial antagonism, when we talk about normal flora, just means the flora that's on your skin is antagonizing anything that's coming in contact with this patch, starving it out, and preventing it from binding onto your cells because it's already there binding onto your cells. That's microbial antagonism. I just don't think I used that term, but that's what it means. Physiological and chemical mediators, temperature. Why does for those of you that have a micro, you get an infection and the temperature of your body goes up, so you get a fever, right? What's the why?
The microbes don't like that, which is
Right, so different microorganisms have different growth optimal temperatures. Those microorganisms that cause infection in the human body like 37 degrees, which is what 37 degrees Celsius is what the body temperature goes up. They don't like that. They usually start to not grow as well. So that's one of the reasons you're getting a fever response. You haven't really got to it yet since we're talking about fever response. Another thing your liver does when there's a fever is it releases something called hepcidin. Hepcidin goes around and essentially binds up any kind of free iron that might be remaining in your body to make sure the microorganisms don't have access to the essential micronutrient. Microorganisms don't have access to iron, and you don't have any microorganisms. They need it to make new cells just like we do. Iron is important for a lot of things. Use it with enzymes to make more glycans, to use it for binary fission. It's important for components of electron transport chains, so that your cells can make ATP, if it doesn't have iron, so essentially they just we'll talk a little bit about how some might around that one too. Lysozyme we talked about, hydrolyzing cell walls that breaks down glycans, you find that in tears, you find other secretions you find as well. High lactic acid, electrolyte concentration in sweat. Most microorganisms don't like high salt concentrations to grow in your body. They also don't like acidic conditions, which is your pH, low pH, and sulfate. Most microorganisms can't survive that. On top of having to do the barrier, on top of having to get around all the normal flora that's there. When you look back at this stuff, after we go through this semester, sometimes you will kind of, I think, be surprised that innovations infections at all because there's so many things that have to go wrong for us. We're one of those microorganisms that cause infection. But it happens all the time, right? Skin's acidic pH, talk about that. Microbial acid stomach, talk about that. Digestive juices and bile of the intestines, same kind of thing. Microorganisms don't like those. Semen contains antimicrobial even though we still do see bacteriological sexually transmitted infections, so obviously that there's problems there as well, or can be. I'll come back to we've talked about microbial antagonism and the female genitourinary tract in here, or did I not talk about that yet? Yeah. Okay. So remember, in the vagina, you're going to have lactobacilli, you're going to have a bunch of different normal flora, but you're going to have lactobacilli, you're going to have sometimes a little bit of Candida albicans, which is yeast. Lactobacilli is the main one that does this, specifically Lactobacillus acidophilus, but there's some other ones as well that keep the pH in that area very low. You take an antibiotic for whatever, wipes out some of that normal flora, including Lactobacillus, pH starts to go up because you know the Lactobacillus they're actually making the low pH, right? When that happens, Candida albicans, yeast, loves neutral pH, start to get a lot more yeast growing, you get a yeast infection. So most pathogens that would infect that area don't like acidic pH. However, if it becomes a neutral pH, there are a lot of things that like to infect. Because most of the microorganisms that infect the human body like neutral pH because of a lot of neutral pH. If we were in acidic pH, there would probably be a lot of microorganisms that like to infect us, but like acidic pHs, but we're not. You have components of complement, which we'll talk about here in a little bit. It's different interferons, which we'll touch on a little bit as well. We'll come back to those. And you also have lactoferrin, so lactobacillus, which is lactoferrin is a compound that binds up iron. Transferrin is another kind of similar component that binds up iron. You find transferrin in the bloodstream usually. We talked a little bit about hepcidin. All these things are things that your body makes to bind up iron, so it's not accessible to microorganisms. Microorganisms want iron, microorganisms don't grow. What happens, unfortunately, sometimes is that microorganisms, actually bacteria specifically, some of them have the ability to make siderophores. Siderophores are secreted by the bacteria. They bind up iron, and then they take iron back to the bacterial cells. Sometimes the affinity for iron by the siderophores are a lot stronger than the affinity of lactoferrin transferrin for iron, so they actually rip the iron away from those cells, depending on the microorganism. We talked about these. Okay. For the phagocytes, we talked about here a little bit. So these are some of our white blood cells that are able to engulf and break down pathogens. Non-specific results in inflammatory response. Neutrophils, dendritic cells, macrophages, monocytes to an extent. Monocytes are precursors to macrophages. Monocytes are immature turn into macrophages. But these are all phagocytic cells. Some of them act as antigen-presenting cells. If you've had an immunology, that's why I refer to as APC. An antigen-presenting cell is something like a macrophage. Takes in a bacterial cell, cuts it into pieces of parts, and presents those pieces of parts on a surface as like little trophies to show the other cells in the immune system what microorganisms they come across. That's the beginning of an adaptive immune response. That's the beginning of a memory of an infection. We'll come back to those. When this macrophage gobbles up the bacterial cell, it gobbles it up into something called a phagosome. Inside those macrophages, you also have lysosomes. Lysosomes are all where all the degradative enzymes are to break down the bacterial cells. When the lysosome and the phagosome come together, they make a phagolysosome, and then the bacterial cell can be broken down. That does not happen all the time. Some bacterial cells have things like capsule that allow it to prevent it from being broken down the phagolysosome and survive inside the phagolysosome, which is a great place for a pathogen to live, because your white blood cell is going to be recognized as foreign by another white blood cell. The bacteria is hanging out there. Right? So there's some pathogens that do that, and we'll come back to those too. Again, pattern recognition molecules, toll-like receptors, PAMs, MAMs, we'll talk a little bit about those as well. What phagocytosis looks like, again, bacteria taken in the phagosome. We have lysosomes, the components are going to break down the bacterial cell. Phagosome and lysosome work together to make a lysosome. Let's say this is bacteria, or you want to read then, bacterial cells broken down, and the components in the macrophage are going to be presented on the surface of the macrophage and then the phagosome. We will talk about this probably through the rest of this week and next week. So different microorganisms have the ability to essentially stop this process at different points. Some microorganisms just have mechanisms that are aided in avoiding being phagocytosed in the first place. Some microorganisms have the ability to survive inside the phagolysosome and hang out in there like we just talked about. Some microorganisms can make different enzymes and things that prevent these two from fusing, the lysosome and the phagosome, so you never get your different things. So we'll talk about some examples of those within the next week. This is what a neutrophil looks like, or a polymorphonucleoleucocyte, or a PMN. You will see these multi-lobe nuclei. That's what a neutrophil looks like. There, here's our process of inflammation. So acute inflammation is just an innate immune response. You're going to see these different parts of that due to the chemical mediators, fever, redness, warmth, swelling, pain, and tissue organ dysfunction eventually if the infection progresses on that. Say you have somebody in this little example here, they somewhere on the shelf. If you see someone that gets, this person's got like a splinter or something, right? What do you think the first thing, if you get some kind of a penetrating wound, what's the first thing your body does due to an injury? Vasoconstrict or vasodilate? Vasoconstrict. If you vasodilate, so vasoconstrict first. I feel like sometimes some of the nuance from this stuff is missed, so I'm gonna go through this. Vasoconstriction first. That injury is gonna induce cells to come along like mast cells. Mast cells are gonna do components like histamines. Histamine's gonna cause Histamine's gonna cause vasoconstriction. Or sorry, I went backwards on it. Histamine's gonna be released to cause vasodilation. You float me up. So vasoconstriction, so you don't bleed out, right? Mast cells come to the area, mast cells release histamine, histamine's gonna cause vasodilation. The reason that you want histamine to cause vasodilation is because a lot of your immune cells are in your bloodstream, so you have to get them out of the bloodstream to the area where the wound is. So you need vasodilation. Because the white blood cells, some of them need to get to that site through a process referred to in immunology called diapedesis. Diapedesis is when these white blood cells squeeze between these endothelial cells into your bloodstream, blood vessels, I should say, and get out to this area where there's a pathogen potential, where there's an injury. You're going to have fever. We talked a little bit about fever. Some of these chemical mediators secreted in these white blood cells in that area are going to cause a fever response. The fever response is going to increase the temperature because you want the temperature to go up because the pathogen is not going to like that increased temperature. That's not going to be the growth optimum temperature. Why do you think, so we're going to go into redness now, why do you think that area is going to get more red? Don't overthink this. Yeah, ultimately, yeah. You have increased blood flow area. Increased blood flow, your blood is red. So it's the iron. You're going to get redness. So if you kind of think about how an infection happens, you can kind of walk through these in your head without actually knowing. You have increased blood flow to the area. That area is also going to become warm, right? So you're going to get redness and warmth from increased blood flow. As those white blood cells go through diapedesis and get out into this area of infection, there's some plasma that leaks out from your blood into that surrounding area, right? So when that plasma leaks out, that plasma is going to increase the amount of fluid in that area, which is going to result in swelling. So that area is going to become red, hot, and swollen. All because of increased blood flow and swelling is from the plasma coming out of the bloodstream. The plasma is going to eventually take it back up through your lymphatic system. The lymphatic system recovers this plasma and dumps it back into the bloodstream. We talked about that during the moment too. Probably not everyone just did, but we'll get to it. The one I feel like maybe isn't explained as much is the pain part. So why do you think there's pain there? From what we've talked about already, not from, say you got stabbed, I know what matters. Why does this response cause pain itself?
Yeah, the increased volume in the area produces more pressure on like the nerves that are being.
I've never had an animal get that right. Exactly. Increased pressure there because you have swelling. That's going to push on the nerve endings. That's not necessarily going to be fine. Good job. So that's how an acute inflammatory response works. So again, without even kind of knowing the details of it, when you think about how the process happens, you can kind of put this together. Obviously, if this progresses long enough, you get tissue organ dysfunction. A lot of these things we talked about are due to different cytokines and interference being produced. Histamine, we talked a little bit about prostaglandins, leukotrienes, complement cascade, we'll talk about in a little bit. But again, make sure you kind of understand this process that is happening. You don't need to really know too much about the chemical mediators. It's not immunology. I just want you to have a general kind of grasp of how this stuff works. So when I talk about some of these infections, to mention different things, you're not lost. Okay. So let's talk about that adaptive immune response a little bit more. So once the innate immune response has been overcome, then you're looking into the adaptive immune response. Again, you're talking about things like T cells and B cells. We talked about that. Actually, one thing I do want to mention on the slides here is there are some things that mucosal associated lymphoid tissue we'll talk about. There are different parts of your body where you have collections of lymphoid tissue that aid in fighting off or preventing infection. We'll talk a little bit about those later on. I'm trying to think if there's anything else I wanted to touch on real fast. Yes. There's a couple of things. Where do B cells mature? Anyone know where B cells mature? Actually, let me go back. Let me get your confusion a little bit. Where are B cells made? Where are they mature at? Bone marrow. And bone marrow too. So usually what people always teach you is where do they mature. So B cells are made in the bone marrow and they mature in the bone marrow. B cells, just remember B cells, remember B for bone marrow. That's not really what it stands for, but if that's what helps you remember, that's cool too. What it really comes from is bursa fibrillis or something, and burrs. It's really, don't worry about it. B's for bone. Where are T cells made? Bone marrow. See, they don't usually teach you this way. T cells are made in the bone marrow, but they mature in the thymus. So that's important to kind of know that a little bit. I'll come back to the spleen because I think I'm going to confuse you. We'll talk about that now. But your spleen does a couple things. One thing it does is it recycles whole blood cells, red blood cells, I should specify that. Red blood cells, as they age, they eject their nucleus. That's part of their natural aging process, and it helps. It's one of the indicators that the red blood cell is getting older and needs to be degraded and new red blood cells need to be made. Your spleen does that. Your spleen also clears bacteria that are opsonized. This is something we haven't talked about yet. So antibody-coated bacteria will get cleared in the spleen. We'll come back to that. The only reason I want to mention the spleen specifically is because there's different types of bacteria that get cleared by your spleen. If you have someone that doesn't have a spleen, they have problems with those kinds of infections. So you can have a couple of different kinds of conditions for people. They can be asplenic, or they can be functionally asplenic. Asplenic means they don't have a spleen. Functionally asplenic means they have a spleen, but it don't work. So we'll talk a little bit about that here as we go throughout the course. Again, there's some specific pathogens. If you don't have a spleen, or your spleen's not working right, you're going to have some problems with it. Adaptive immune response, specific to particular antigen. So again, this is your specific immune response. So it's going to recognize particular things. Very specialized. It's expressing immunological memory, so it's going to have an immunological memory of previous exposure to different pathogens. It's capable of self versus non-self recognition, which it's supposed to be. Sometimes you have somebody that has an autoimmune disease. That's what is wrong with that. It's recognizing self as form. It's self-limiting. It's supposed to return to a basal resting state after whatever immunological challenge it was exposed to. Again, sometimes some problems with that, but it's supposed to not overreact. Eliminates the antigen, hopefully, and then you have a long-lasting protection against that kind of infection. But one thing to talk about here over the next month or so, people ask me sometimes, like, why if I had strep throat and I had an immunological response to it, why did, like, 3 months later I got strep throat again? The reason is there's about 23 different strains of strep throat. So unless you had all 23 at the same time, you're probably going to get strep throat again. So there's different ways that some of these get around it. Antigens, so this is important to kind of note here too. So your immune system's going to recognize antigens. The best antigen that your immune system's going to recognize are proteins. Other things that don't recognize real well. DNA, lipids, phospholipids, sugars, carbohydrates, things like that. Your immune system really does not recognize well. It does kind of a crappy job of recognizing those. But proteins it tends to recognize really well. If you have a protein, there might be multiple antigenic determinants on that protein. So an antibody might recognize a little component of that protein, and another antibody might recognize another component. So that's all to say you might have one protein, but that one protein might have 8 different antigens on it that 8 different antibodies can recognize. So antibodies are going to recognize really tiny parts of like a protein, not the whole protein. Sometimes you'll see antigens referred to now as immunogens. Antigens was the term we used to use, essentially describe things that are recognized by antibodies. However, antigens can stimulate more than just antibody-mediated immune response, so human immune response. So now we refer to them as immunogens. All means is something that triggers an immune response. That's an immunogen. Macrophages and dendritic cells, again, can act as antigen-presenting cells, present whatever they've created to other cells. We'll talk about that a little more specifically in a moment. The components of your MHC are made of histocompatibility complex. There's actually 3 classes we want to talk about too. So all nucleated cells in your body are going to be class 1 MHC protein. And those class 1 MHC proteins are going to take a non-self antigen, so like say we're going to go to the bathroom, and then present it on the cell surface. So when I say all nucleated cells, what cells in your body, I told you this before, what cells in your body will increase MHC class 1 on your surface? Red blood cells. Even platelets make red blood cells don't. Class 1 MHC proteins tend to present things on their surface that are foreign, that are considered, and I'll talk about this a little bit as well, so don't worry about it at the moment. They tend to present things on their surface that are endogenous. So say you have a cell, one of your cells that has a viral infection, they tend to present things like that on the surface. Sometimes you have intracellular bacteria, they would present components of that on the surface. When you talk about MHC class 2, those are found on the surface of B lymphocytes, macrophages, and dendritic cells. Those tend to present things that were exogenous, so outside the cell. So B cell macrophages and dendritic cells, albeit with different mechanisms, are going to bring the pathogen in from the outside into the cell, break it down, and present it on the surface. So class 1 tends to be things that are already in the cell that's presented on the surface. Class 2 tends to be things that your cell brings in, chops up, and puts on the surface. Does that make sense? It might be a little bit, if it doesn't make a little more sense, we can go through all this. For your MHCs, don't worry about the tissue distribution besides MHC class 1's on nucleated cells and platelets, and MHC class 2 or more on phagocytic cells. You don't need to know all the names. Don't worry about this. All the HIV, you don't need to know. MHC class 1 is recognized by a specific type of T cells called cytotoxic T cells. You'll sometimes see those abbreviated as TC cells, and C is the subscript. You also see with a lot of these as you go throughout, they'll have next to them and say like, I'm trying to sorry y'all. For those of you that have immunology, I'm trying to make this really simple for those of you that have not had immunology, so you're not super overwhelmed. Sometimes with sometimes, holy God. Sometimes for these different white blood cells, you will see them referred to by CD and then some number. What that refers to is cluster of differentiation. All that is a protein that's on the surface of these cells. And you can use them to differentiate the different types of white blood cells. Different white blood cells are going to have different clusters of differentiation on their cell surface. You can use them to distinguish down apart. That's all this means. Cytotoxic T cells have a specific CD, CD8. So there's CD8 plus means they have. CD8 minus would mean that they don't have it. But all cytotoxic T cells are going to have that CD8. TH cells, also referred to as helper T cells, have CD4 on the surface. CD4 is going to recognize MHC class 2 on your cells, on your phagocytic cells. And cytotoxic T cells are going to recognize MHC1 on all nucleated cells. So we'll go a little bit more detail about this here shortly. Again, MHC1 is going to usually present things endogenously synthesized, and MHC class 2 is going to present things that are outside exogenously processed. Function for MHC1 is to eliminate abnormally infected host cells by cytotoxic T cells. Sometimes there's a little bit of a ability of these cells to also recognize malignant cells as well, cancer cells. Obviously, that sometimes will work. For MHC class 2, function present foreign antigen to helper T cells. We'll talk about this here a little bit when we have some figures. Okay. So, MHC class 1. This is a cell you know has a nucleus, so it's pretty much it could be any cell, but it's not a red blood cell. There's something in here, say this is some kind of a virus. That's the example that what other things would count. There's a virus in there. Where do you find viruses in your body? Inside your cells. Because can a virus survive without being inside your cells? Because what does it need? It needs, yeah, it needs everything pretty much. I mean, so a virus is going to be some genetic material that could be single-stranded or double-stranded RNA, single-stranded or double-stranded RNA, DNA or RNA, little URL, and a viral coat on the outside of this has a capsid. Well, it can't make more of itself. So to make more of itself, it needs to hijack your host cell machinery like your ribosomes to make more of itself. So it has to be, by definition, to exist inside your cells. Unless you like sneeze something out to pass to somebody else to make more of itself, it's going to be inside your cells. So that's an endogenous, it's inside your cell. So a component of it can be presented with MHC class 1 on the surface of those cells. So that's endogenous. Again, there are some bacteria that prefer to live intracellular. Those ones kind of have the same mechanism. MHC class 2, something's taken out, like a macrophage. It's gobbling something up that's outside, bringing it in, breaking it down, presenting its components of that pathogen on the surface of MHC class 2. So that's an exogenous pathway. Usually. So we'll talk a little bit about those as we move through. For your humoral immune response, your B cells, they're going to mature in the bone marrow. They're made and mature in the bone marrow. They don't really move. They're not those type 1. They're activated by interaction with a helper T cell. It's a type of helper T cell, a T helper 2 cell. Differentiate then into plasma cells. Plasma cells secrete antibodies, and then also memory cells, which have immunologic memory of that antigen, whatever it may be. The antibodies themselves are also referred to as immunoglobulins. They're Y-shaped lipoproteins that look something like this. That do not be known in great detail. and there's some sugars and stuff. Whoop-de-doo. So, what did I want to tell you about that? Oh. So, your Y-shaped antibodies are going to be divided into two regions. This region up here is going to be your variable region, and this region down here is going to be your constant region. This constant region down here can be recognized by some of your white blood cells that have FC receptors. Sometimes this constant region you see referred to as FC, whatever. You'll see this referred to as FAM. You don't really need to know any of this. But down here, say you have this, let's say this is a macrophage. You have a macrophage. It can have an FC receptor. You have this antibody that's out wherever, recognizes an antigen, antibody binds on to this FC receptor through its constant region, and then can take that antigen, that antibody binding antigen into the white blood cell, wherever it may be. There are different microorganisms that can make different proteins that block this constant region, so they block this interaction. So you can't take them up because there's not that interaction. There are some bacteria that just secrete different proteases that degrade these antibodies. There's all kinds of different virulence factors. Up here, just remember also, every antibody is going to bind two antigens, but it's always going to be the same antigen. So what does that mean? So you have this goofy looking protein here, and the antigen you're interested in is right here. We're going to call that antigenic determinant A. This will recognize this. Let's draw the same protein over here very poorly. This side will also recognize that. We're going to recognize the same thing twice for each antibody monomer. That'll make sense when I'm specifying that here in a moment. But sometimes people get confused on, is you have, say you have something else over here that's a different antigenic determinant B, and be like, oh yeah, it can recognize two things. So like this, we can then recognize A, and it can recognize B. No, that's not a thing. They're always going to be the same thing. It's going to recognize the exact same thing. Now, can you make an antibody in the lab, like using like a hybridoma or something to recognize two different things? Yeah, you can do that. But your body doesn't do that. It's going to recognize the same thing. I went a little Rain Man on y'all there, sorry. I don't know what that was about. I'm going to start drinking coffee apparently. Let's see. What else? There are different types of antibodies. We'll talk about those in a little bit. Some antibodies exist as this, just like a monomer. Some of them exist where you have more than one of these put together. We'll talk about that in a little bit. The primary response to an antigen is going to look something like this, which I usually show you this figure and then redraw it on the board as well because I'm a psychopath apparently. I don't know why I do that. You have a B cell. Actually, I don't want to use this example. Let's use a macrophage. Say this is a macrophage. This is a macrophage. You have a macrophage. Let me tell you why. So B cells are not phagocytes. The way B cells recognize antigens is B cells have antibodies on their surface. They recognize something is formed through that antibody, and then when that antibody binds antigen, it induces the B cell to take that antibody antigen inside the B cell. So you have to have that antibody for a B cell to engulf it. Macrophages are just recognizing something like through a toll-like receptor, say lipopolysaccharide or something, to take it in. So the kind of more classic way to explain this is not to use, where I put B on this 90 times, not to use a B cell, to use a macrophage. It's just a little bit easier to explain. So macrophage, say, engulfs some kind of a microbe. What does it engulf it into? What's that organelle called? Macrophage engulfs it, and it's inside of what? Phagosome, good. Phagosome is gonna merge with the lysosome, make a phagolysosome. Microbe's gonna be broken down. Microbe's gonna be presented on the surface of the macrophage using MHC class 2. Because this was brought in from the outside, right? So it's brought in from the outside, so it's an exogenously processed microorganism. Broke it down, presented with MHC class 2. So now on the surface here, macrophage, you have presented pieces of this microorganism with MHC class 2. MHC class 2 with some kind of antigen bound is going to be recognized by a T cell receptor. T cell receptor is the receptor that's found on the surface of T cell. T cell receptor is going to recognize that. I'm going to draw this because you can't see that with a magnifying glass. My 3 inches of real estate on this dry erase board I have. MHC class 2 is going to be recognized by a T cell receptor. Let's do this. T helper. Macrophage. So this is your MHC class 2. Let's say this guy is your antigen. And this is your T cell receptor. T cells, helper T cells have CD. Anybody remember that? From a couple slides ago, CD4, good. CD4, how I usually explain it, is it kind of acts as like a double check here. So it's making sure that this interaction is actually supposed to be happening or not. That's on the surface of helper T cells. If you have this interaction, it's going to stimulate different things to happen, which we'll talk about here in a little bit. If that interaction is with a B cell, one of the things it's going to do is it's going to stimulate the B cells, differentiate into plasma B cells make secrete antibodies. It's also going to cause some of that B cell population to turn into a memory B cell. Memory B cells are going to have an immunological memory of whatever this antigen was. There's a specific type of helper T cell that is involved in aiding the maturation of B cells. There's another subset that's involved in doing this kind of interaction that actually is involved in causing stimulation and activation of macrophages. They're different subsets, but they're both helper T cells. They just help in different ways. This interaction that's up here on the slide is Th2, so T helper T cells. Th2s, these are Th1s. I almost told you don't need to know that. I'm pretty sure I'll ask you that, but double check with me before we get that good. Because I don't think I'll ask you that much detail. Yeah.
So if a macrophage takes up the microbe, processes it into an antigen, and shows it to a T helper cell. So then that T helper cell takes it to a B cell, or?
So the different subsets of the T helper cells.
Okay.
So T helper 1s, like I drew on the board, is going to activate the macrophage. T helper 2s are for the B cell maturation.
Okay.
Usually, at least I've found in the past, it's a little bit easier to explain this Th1 kind of interaction before I go back. Because you don't need the antibodies for this interaction, but you need it for this interaction. It's just different subsets of T helper cells. They're both CD4 positive.
And then one more question.
Yeah.
So when the macrophage is inactivated, does it present the MC class 2 or MHC class 2, or does it still present that, or does it have to eat a microbe and then process it?
I know where you're going. Well, so a couple things. So it will present MHC class 2 once it has an antigen but it's always going to be presenting MAC. This is what I did one. It's always going to present MAC class 1. Okay. If it has something to present for MAC class 1, which means you have to have macrophages infected with something endogenously, like a virus. Okay. So that's where the nuance of this gets a little, you don't need to know this much detail at all in your immunology, but if you have a macrophage that is endogenously infected with something, it can present through MAC class 2 because it's endogenously infected with something. It can get a little complicated there. Because the nucleus itself, macrophage out of nucleus, perhaps a nucleus, macrophage out of nucleus, that word that right there, third time in German. But, yeah. Okay. Does this kind of make sense? Okay. So, the secondary response to antigen. So if we go back to the slide here, this is your primary response. We're talking about B cells. To get these B cells activated, you have to have this interaction between the MHC class 2 and the T cell receptor, CD4, and all this has to happen. And then it causes the B cells to mature and differentiate into memory cells and plasma cells, blah, 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 blah. Secondary response, you don't need this. You don't need any of this interaction. B cells are going to have antibodies on their surface, the memory B cells. The memory B cells are going to recognize that antigen that it's seen before through those antibodies on the surface, and it's just going to stimulate those B cells to make more plasma cells and more memory B cells. So this secondary response, the second time you see this pathogen or this antigen, whatever it may be, your immune system's going to move a lot faster because it doesn't have to muck around with all this stuff here. So if you look at that, your antibody production, that's what this looks like. So if you're exposed to some kind of a pathogen down here, and this might not make a ton of sense here for another few slides, you're going to see a spike in antibody production after, it's usually about 7 days or so. Make sure that, okay, good. Y'all can see this. I usually show this graph a lot smaller and you can't see anything. The first antibody you tend to see made is IgM. Everybody remember how many antibodies are in IgM? 5 antibodies. So that means that there's 5 Y-shaped antibodies stuck together. It's a pentamer. How many antigenic determinants does it recognize? Because each one recognizes 2. 2 times 5 is 10, right? You usually see IgM made first. This is kind of evolutionarily associated response because likely IgM is the first one your body makes because it can recognize 10 things instead of 1. So you see this made first. And then you see a spike about, depends on the person, and it depends on the type of infection. Within about 3 to 7 days, you tend to see this spike here in IgG. IgG is a lot more specific than IgM. Long period here until you start seeing antibodies made. Down here, you're exposed to whatever this pathogen is the second time, antibodies are made right away because you don't have to goof with all this stuff. Your secondary response, you're going to see IgG made first because it's more specific. That's the blue line. You also see a lot more overall antibody made. That's what the purple line is than you do during the first exposure. And you have some IgM made as well, but you make more IgG during the secondary exposure. This is a lot more specific. Okay, here we go. Human body makes 5 different types of antibodies. IgG, IgD, and IgE are all on one line. IgA and IgM are the ones that have only one. IgA has 2, so it's a dimer. Each one recognizes 2. So each IgA dimer is going to recognize 4 antigens, or the same antigen, I should say. And IgM we just talked about. IgM is a pentamer. It's going to recognize 10 things, 2 on each one, 5. Those different components of the antibodies for IgM and IgA are held together through So the antibodies, IgA on top of that has something that aids in its ability to stay soluble, called a secretory component. You find IgA secretions like breast milk, etc., etc. We'll talk about that here. So let's talk about IgA and IgG first. IgG, what do I want you to know about that? IgG is the antibody that's involved in conferring resistance, I should say immunity, from mother to fetus, because it's the only one of those five that expresses itself. Long-term immunity, memory, antibodies, neutralizes toxins, opsonizes, fixes complement. We'll talk about all those things here in a little bit. IgG out of all these is the most specific, and IgG is the only one that can cross the whole cell. That's pretty much what you need to do about that. In your serum, it's about 80%, so it's the one you find the most. IgA, you're going to find in mucous membrane, breast milk, and secretions. It tends to be the most abundant antibody in the body. So if you're looking at overall in the blood and the tissues, like in secretions everywhere, you have more IgA than anything else. If you're only looking in the bloodstream, IgG rules. That's where you find the most of in the blood. IgG highs in blood, IgA highs in blood. IgM we talked about being the first to respond to antigens, that's the one that's named the most. It can also serve as a receptor on B cells, so it can function as a B cell receptor, so it can be that antibody surface of a B cell. IgD can also function as a B cell receptor, so it can be that antibody on the surface of B cells. We don't know as much about IgD. IgM seems to be a lot more important than IgD, but both M and D can be on the surface of B cells to recognize antigens. IgE is the one that's a little bit more interesting sometimes. You find it associated with mast cells and basophils. The reason for that, I'll get to here in a moment. I'll flash forward on here a little bit. Couple times. There we go. IgE in the United States, you will tend to see associated with an allergic reaction. You tend to find IgE associated with mast cells. Mast cells, we talked about acute inflammation. Mast cells secrete what that cause vasodilation? Histamine. If you are allergic to pollen or something, you take an antihistamine. It's usually associated with histamine responses. If you go to a foreign country where their sanitation system is not necessarily as good as the ones we're lucky enough to have in this country, you will see different kinds of protozoan and helminth diseases usually associated with fecal oral route, which means there's one of these organisms that lives in associated with feces secreted by us as a vertebrate mammal and poop. And then there's a contamination of that sewage source into drinking water and someone consumes it. In those circumstances, if you're talking about something like a macrophage, obviously not to scale. Little macrophage, if somebody has this large worm infection, there is no way if this little guy is going to gobble up this big worm. So what happens is IgE recognizes this large thing that's too big to engulf through phagocytosis. The constant region of that IgE is recognized by constant receptors on things like eosinophils, and it triggers them to release things that kill this thing from the outside without having to engulf it. So things like perforins. Perforins literally perforate. So they make, essentially, it's the death by like 1,000 paper cuts. Essentially, they just poke little holes in the worm until the worm is no worm. anymore. This is how you see this work outside the United States in certain places in the world. But again, in the United States, what tends to happen is IgE recognizes something it's not supposed to, like pollen, and causes something like a mast cell. This constant region on the IgE will recognize a constant region receptor on the mast cell, triggers the mast cell, or stimulates, as you say, the mast cell to secrete histamine. Histamine causes an allergic reaction. So IgE in nature seems to be associated with like trying to get rid of like big parasites. In the real world here, at least in the United States right now, you tend to see more as a nuisance than anything, because you don't see these types of infections here. If you went somewhere else and were exposed to one of these types of pathogens, you might have that kind of response, but you probably not going to have it. Okay, so the different ways these antibodies can work. You can have neutralization. Neutralization is a real easy one to understand. You have some kind of pathogen. It has a mechanism on the surface, say it's a pilus or something, that's going to allow it to attach to our cells. And what the antibody does is it coats that bacterium, say coats that pilus, so that the pilus can't interact with our cells. So it blocks interaction and neutralizes it. Antibodies bind onto the antigens on the microorganism, prevents the microorganism from attaching to us. The microorganism can't attach to us, it can't establish an infection. Sometimes you will see these referred to as antitoxins. Someone gets bit by a snake. There's the toxin from the snake is in the bloodstream. They're given an antitoxin. That antitoxin is an antibody. The antibody binds onto the toxin, blocks the toxin from attaching onto whatever receptor it does in our body to cause that, whatever that reaction may be. It's neutralization. It literally neutralizes. Agglutination is another one that's easy to understand. Agglutination binds a bunch of the pathogens together, the antibodies. Because if you're a macrophage, it's a lot easier for you to go and gobble up this big clump of bacterial cells instead of trying to go chase down 10 of these. Does that make sense? It's just easier. We talked about this, so that antibody-dependent domain cytotoxicity. Antibodies binding antigen enhances complement. Complement proteins readily form membrane attack complexes and destroy bacteria and enteroviruses. So the binding of antibody to pathogen, to antigen, attracts components of complement, which are small proteins. The components of complement can assemble into a membrane attack complex. Membrane attack complexes are like little pore structures. So what ends up happening? What ends up happening is you get a bunch of little pores stuck into these bacterial cells until they lyse. This works for bacteria. It also works for enteroviruses. So some viruses have a capsid where it's just the naked virus, where it's just the protein on the outside. Some viruses have an envelope, so they have a membrane on the outside of them as well. Usually it's giving into our cells, because the membrane is usually from our cells. So that's an envelope. So if you have that membrane on the outside surrounding the virus, you can poke holes in that envelope with complement, with membrane attack complexes. If you poke holes in that membrane, then it's going to hamper the ability of that virus to actually get into our cells, because you can damage that envelope. So complement fixation works for those things. Oxidization is one of the things we've talked about before. Microbes and foreign particles are coated by serum components in preparation for ingestion by phagocytes. Again, if you have someone that doesn't have a spleen, they're more susceptible to encapsulated bacteria, because that's where the spleen is usually where bacteria that have capsules are sent to be cleared. If you don't have a spleen to clear them, you can't clear them, and you will have an infection. Strep pneumonia, we'll talk about all these this semester. Strep pneumonia, Serocontagious, Moccus influenzae. The ones you tend to worry about here are ones you find associated with the respiratory tract. cause a respiratory tract infection. They can move to the blood-brain barrier, cause inflammation there, and ultimately end up causing meningitis. People that are really splenic oftentimes have problems with bacterial meningitis. Sometimes when we'll talk about this, I think in the spring, they have problems with salmonella as well, because salmonella makes a capsule. But most times it's these kind of ones that cause meningitis all the time. The way you get around this, because so they say splenic, is you immunize them for everything humanly possible that has a capsule you think they might be exposed to and get an infection from. So you vaccinate against all those things. So they get it, but it does not go well. Different kinds of opsonins, antibodies, IgG being the most important one, you can remember some the complement system itself, certain proteins, about 30 of them or more, act as opsonins, coat the bacteria, can cause lysis, but makes an membrane attack complex. The complement system kind of bridges between innate and adaptive immunity. And these are the components of it that you don't need to know. What I will stress upon you, though, to be somewhat familiar with, because you will see this if you sit for an kind of very bad or a board exam or a professional exam or board exam here. They tend not to test you all on all the little individual components. They tend to test you all on just the break points. This person comes in and they have this type of infection, and they have a genetic deficiency in C3 convertase or C3b. Usually they really love to ask questions about C3b. It's just like, well, can they do anything with, like, can they make membrane attack complexes? No, because they can't make C3b, so it shuts this down, shuts that down. They're more interested in asking you all about break points in these pathways, where there's some that do one thing, if you goof it up, it goofs up a bunch of things down, they love asking those questions. So they usually focus right here. I've seen more questions about C3b over the years than I have hairs left on my head, which isn't telling you as much as it used to. But they love asking about that stuff. For cell-mediated immunity, we talked about this a little bit. T cells, mature in the thymus, they're made in the bone, they mature in the thymus, they're mobile, and we talked about the interaction between MHC and T cell receptors. Major role B cell activation. This is kind of some of the stuff we talked about before. There are some different subsets of T helper cells. All the helper T cells that are going to have CD4 as a co-receptor, they're all going to recognize MHC class 2 proteins. However, Th1 helper T cells are going to stimulate inflammation in other immune cells like macrophages. You know, what they'll actually stimulate the macrophages to do is to be essentially increases their phagocytic ability, so the ability to phagocytose quicker. And it also will aid in stimulating monocytes and maturing the macrophages. Th2 helper T cells are going to be the ones that stimulate the B cells to differentiate into member B cells and plasma B cells. The cytotoxic T cells are going to have CD8 as the receptor, co-receptor, and they're going to recognize MHC class 1. So this shows you the same thing that we talked about. So you can look at that if it helps you a little bit. This is showing you those interactions. This is showing you those interactions again. We haven't talked about this one as much, but this is if you have, say, a nucleated cell and it has, say, a viral infection, presents with MHC class 2, cytotoxic T cell that has CD8. The CD8 and TCR is going to recognize the MHC class 1 bound antigen, and it stimulates the cytotoxic T cell to release things that are going to kill that diseased nucleated cell, whatever that nucleated cell may be. For your specific immunity, you're going to have specificity, so it means you're going to recognize reactive individual molecules via direct interactions. It's going to have memory, have a molecular memory of that previous exposure, and it should not recognize self-antigen, even though sometimes there are different disease processes that where that does happen, unfortunately. Innate versus adaptive. Phagocytic cells are processors and antigen, we talked about that. Cytokines can be released by T cells, all the stuff that I talked about. But all of these next couple slides are trying to show you is essentially the components of required innate immunity do not just operate as separate things. They communicate with one another. This shows you the exact same thing. And then lastly, and then I promise you will take a break. You require immunity in different ways you can get that. So you can have two different types, and there's some subsets of those. You can have natural immunity or artificial immunity. Natural immunity is what it sounds like, you get that naturally. Artificial immunity is what it sounds like, you get that artificially. There are two types of natural immunity. There is active immunity and there is passive immunity. Active immunity is you get sick, your immune system responds to it, and your immune system has an immunological memory of whatever that infection was. That's natural active immunity. Passive immunity, natural passive immunity, is you get antibodies from, say, your mother. That can be through breast milk, so IgA. It can be through, as a fetus, breast milk placenta, IgG. Those antibodies, it's a passive immune response. Those antibodies do have a half-life. They usually last months, not years. And when they're gone, they're gone. Because that baby has never been exposed to whatever pathogen caused the mother to make those antibodies. So once those antibodies are gone in the breast milk, and they're exposed to that pathogen, they're going to get sick from that pathogen. Because they don't have any immunological memory. They didn't do that on their own. They were just using antibodies passively for the mother. So those are the natural examples. The artificial examples, artificial immunity, are artificial active immunity, which is you go get a vaccine. That's why nobody in here has polio. You go get a vaccine, it's got to stimulate immunological memory against the polio virus. You should get polio. So that's artificial active immunity. Artificial passive immunity. Excuse me. Artificial passive immunity is when you get antibodies given to you. So anti-serum. What would you use that for?
Snake bite?
Snake bite, venom. Give somebody antitoxin against that snake venom. Antitoxin is antibodies. We looked at this stuff for COVID-19. So people would get antibodies from other patients that survived COVID-19. Those studies kind of go back and forth. I remember some of the ones we did, they seemed like they worked, and then you kind of run some other trials that seemed like they didn't work. I don't know. Sometimes this stuff works, sometimes it doesn't, unfortunately. But it works for snake bites. The same kind of thing. Has anybody ever heard of Kawasaki's disease? Kawasaki's disease is a juvenile disease. It causes inflammation of the vasculature. We don't really know what causes it besides it is happens in kids sometimes, and it causes inflammation of the vasculature. Congratulations, I told you everything we know about Kawasaki's disease. We don't know if there's some kind of virus that we haven't detected or something that causes that inflammatory response. We just don't know. What we do know is if you take serum from, say, other kids and give it to a Kawasaki patient, the inflammation goes away. So what the thought is there's something that people are exposed to and then end up making antibodies against it that a Kawasaki's patient can't make for some reason when they're exposed to that virus. So if you give them someone else's antibodies, but I mean, it's just, we're kind of guessing, to be honest. We don't know, we just know that we give you the serum, the anti-serum, and that works. But we don't know what the antibody is actually detecting. Or maybe it's something else, but I mean, that's kind of the general thought process. It must be something that caused some kind of infection that's blocking it. We don't know. So do you think artificial immunity or you think passive immunity? We think more neutralization that is a long time. So you just teach it on passive. True. complete, so we'll do